we are going to get started with our first invited talk of the day. We have Dr. Kenneth Blum. He's a retired full professor of pharmacology and chief of the Division of Drug Abuse at the University of Texas. Dr. Blum will be talking about reward deficiency solution system, neurogenetic and neuroimaging, translational neuroregulation having clinical relevance. Please welcome Dr. Blum.
Okay, so this is about a 16-year follow-up, I imagine, since the last time I was here. As you can see, I'm not a tall Texan, so I'm from New York City, so I can ask everybody, how you doing? All right. All right. So we're gonna try to, in the time allotted to me, go through about 50 years of research related primarily to addictive behaviors. And what I'm going to do is I'm going to actually come on downstairs and talk to you from downstairs because I kind of don't like the podiums. So let's start and do that, okay? Yeah. Make life easier for us. Okay. All right, now I got to just check this out and make sure that it works. Oh, it does work, okay. Excellent. But it doesn't go back. Yeah, it does, okay. Okay, so we're going to talk to you about reward deficiency solution system. I want to remind everybody that if you have any interesting papers, I'm the editor-in-chief of the Journal of Reward Deficiency and Addictive Sciences. We'll welcome the papers, and that would be kind of nice. I'd like to give you my conflict of interest. I hold patents on something called KB220Z. I hold patents on the genetic addiction risk score, and I license out products to LaVita RDS and Victory Nutrition International. So with that said, let's see what we can do about bringing about some excitement, okay? So in 1978, we came up with something that we call the types of addictive craving behavior. And what we really have here is 3 different types. And we have you're born a basically addict, and that's the craving behavior is equal to the genetics that bring about a deficiency of neurotransmitters plus the environment. The second is that the genetics is okay, but the stress induces a deficiency in neurotransmitters. The third type is when you start using drugs, that the genetics may be fine, but you get a deficiency of the neurotransmitters. So that you can imagine if an individual has genetics, lives in an environment, has stress, starts using drugs, now you got a real problem on your hand. You got a problem, any one of those are a problem, but that's what you normally see out in the field. Interestingly, a lot of the addiction world considered that it was basically a psychological phenomenon, but the Addiction Society in Medicine in August 15, 2011, said that addiction is a primary chronic disease of brain reward, motivation, and memory, related circuitry, dysfunction, and these circuits leads to characteristic biological, psychological, social, and spiritual manifestations. This is reflected in an individual pathologically pursuing reward, and that's important, and or relief by substance use or other behaviors. So that sort of gives you the idea of what we're really talking about. Now I'm going to ask some questions and I'm going to give you some answers. What is the difference between reward deficiency syndrome and addiction? So reward deficiency syndrome is a relative failure of the dopaminergic system, which plays a major part in brain reward mechanisms. The syndrome has been linked to dopaminergic dysfunction, acute excess of chronic deficit of dopamine release in the brain reward circuitry. This deficiency includes various conditions, such as alcohol abuse, smoking, obesity, pathological gambling, ADHD, okay, in which the subject seems to be unusually concerned to achieve reward. Finally, polymorphisms, these are gene variants of a number of reward genes, including serotonin and the cannabinoids GABA, glutamate, and dopamine have been correlated with a chronic dopamine deficiency and reward-seeking behaviors. Also includes the opioids, of course, as well. Now, if you take a look with the DSM-5, we kind of designed this out of the DSM-5, and you notice that reward deficiency syndrome is involved in addictive behaviors, substance and non-substance related, impulsive behaviors, obsessive compulsive behaviors, and personality disorders. As you can see, it's an array of a lot of these behaviors are hooked together under one umbrella. Now, when you look at it, you might say, my God, that's a very long, remarkable list of a lot of stuff. But we promise you that that before the day is up, you'll understand that this is a list that truly reflects what's going on. And that's the reason why we have so much transfer of addictions from one addiction to the other. And I'm sure you see that in your clinic. Now, the difference between reward deficiency syndrome and addiction. All addictions have a common neurochemical thread, which is both genetic DNA and epigenetic RNA environmental. Dopamine deficiency is the common neurochemical pathway. That is how transfer of drug and non-drug addictions occur. Non-drug addictions include behaviors such like food, sex, gambling. Substance and non-substance addictions are a subset of RDS, okay? Okay. Now, what we're going to be talking a lot about is the molecule dopamine. Now, just quickly, you might say, but why are we just talking about dopamine? We're not just really talking about dopamine. We're talking about the net release of dopamine through the brain reward system, which we'll explain to you what that's about. But this is an X-ray crystallography of dopamine. As you can see, it looks like pretty happy molecule. Now, so how does dopamine get released in the brain to enhance well-being, motivation, and reduce stress? Well, if you take a look at the brain reward circuitry, what you can see is that there's a whole bunch of areas. I'm trying to use this pointer, but let me see if I can get it. Yeah, there it is. So everything from the prefrontal cortex, involving the thalamus, the limbic brain, nucleus accumbens, the VTA, which involve medulla, and the hippocampus. So it's a very complicated but very well-known researched area. I'm sure most of you know all about it. Now, this is something that we developed with Joe Kozlowski. I don't know if he's here, but we developed this in 1989 called the Brain Reward Cascade. And what we did is we were able to kind of say that in the hypothalamus, serotonin 2 serotonergic receptors can affect the mu-opioid receptor, where we get a caffeine release, which that can affect the GABA receptors in the substantia nigra. And that GABA-A can affect the raphe nucleus, which is going through here, through the ventral tegmental region, which is glutamate. And once that happens, it then projects out into the nucleus accumbens, we end up with dopamine release. So it's a cascade of events. Okay, so but the end result is dopamine. At this point, we don't know anything else after dopamine. If you know something, let me know, and I'm glad to change my mind. Okay, so now, what we have to consider when we're dealing with patients, whether they're coming with ADHD, eating problems, if they're coming with drug addiction problems, gambling problems, which, by the way, I feel, and I'm going to tell you at the end, I think that neurofeedback has a great relevance to all of that. What you have to take a look here, though, is to see what we consider a happy brain, which is this, and if you take a look at it, you can work. Stop working. Okay. It doesn't work. Oh, there it is. So we get the happy brain, and notice that we have this brain reward system working properly. Everything is working in tune. dopamine is being released at the right amount. In the unhappy brain, you might have too much GABA, which will hold back the dopamine, right? At the nucleus accumbens to the VTA, to the ratified nucleus, and you get a little dopamine coming out. Now that can, in a sense, trigger what we call anhedonia, or maybe depression, okay? If you got too much, you might have schizophrenia. So this system is very important and very involved. Okay. So dopamine is released into the synapses of the brain to enhance well-being, motivation, and reduce stress. So a neurotransmitter is a chemical messenger which carries messages between neurons. Dopamine is a neurotransmitter. The interaction of neurotransmitters like serotonin, cannabinoids, endorphins, GABA, glutamate cause the net release of dopamine in the reward site of the brain, which is the nucleus accumbens. Stress induces the release of the stress molecule, norepinephrine, and dopamine blocks its effect. Dopamine is released 100 times above rest to block norepinephrine effects when you have stress. The release of dopamine also causes feelings of well-being, motivation, and pleasure. That's the thing that's going to make us happy. Okay? Now, so what are the neurochemical common mechanisms between drug and non-drug addictive behaviors? What are we talking about? So some years back, I'm talking now 69, 70, we started to look at the possibility that naloxone, which was an opiate antagonist, okay, would actually block the effects of ethanol, and absolutely we showed that it does that, okay? So, and that's, remember, many, many years ago. And then we started to look at genetically bred mice, okay? And we realized that genetically bred mice, some of them drink a lot, some of them drink in between, some of them don't drink at all. And what we found is that's related to the basic whole brain and also selective regions of methencaphalin in the brain. So if you have a high methencaphalin, you're not going to drink a lot. You got low methencaphalins, that's when you start drinking. So low endorphins, low methencaphalin increases drinking behavior. That's very important. Now, in 1982, we published in Science that if you think about the third type, so where drugs can affect the brain and can affect the chemistry of the brain, we showed that in golden Syrian hamsters that love to drink alcohol, after 1 year, when we look at the endorphins in the basic ganglia of the brain, what happens is that it blocks the synthesis. That's a normal hamster. We had 7 of them, happy, dopey, you know, all the 7 dwarfs, and Ken was one of them. And then you can see that this what happens to a, of course, this is one animal, this is a control animal. This is the control animal, this is the animal drinking after about 20. It's like 20 years of drinking alcohol, and you can see it just wipes out the endorphins. And you can see that's another one, that's another one. And actually it's been shown that morphine does that, benzodiazepines do it, and cocaine, by the way, has recently been shown that it can do this. It can knock out the endorphins, the natural opiates. So too much of a good thing can be toxic. That's what my mother used to tell me, I don't know why. But anyway, can an encapsulinase inhibitor reduce craving behavior? So can you inhibit the breakdown of encapsulins, okay? And if you do, you raise the encapsulins up, and what should happen? You should have a reduction, right, of drinking behavior. So we did that, and what we show here basically is that if you have animals drinking, you give, that's saline. Saline out of stress knocks it down, but when you give CSF, they get a little used to it. Then you give the encapsulin and the bango, the drinking goes down, the water levels do not change. So kind of showing, yes, the concept that encapsulins actually affect drinking behavior, okay? Now we decided to do something else. We wanted to see if we used a substance called diphenylalanine. which is a carboxypeptidase inhibitor, has been around since 1933, and they know that, okay? And this substance actually blocks the action of the enzyme enkephalinase onto breaking up enkephalins. What we're able to show is that we can actually increase the enkephalins, and in this particular experiment, we show that stress, when you have stress, if you had this room where you're separated, one side we give stress, okay, because you're swimming cold. This side you're not having stress, and when you take the animals out of those conditions, the control animal goes to tea, and the other animals start drinking alcohol. And this is due to a change of enkephalins in the brain, and you can see that change occurs after the swim coach. You can see it starts going down. Now, what we've done is say, well, if we raise the levels with the diphenylalanine, what happens is that you could do what we call a pharmacogenetic engineering, meaning we could switch the activity, the behavioral activity of one animal, and make that animal look like exactly like another animal. And what we mean by this is that if you take a look at the yellow one is a C57 black mouse, and the C57 black mouse loves alcohol. If we give them 18 days treatment of diphenylalanine, the acceptance of alcohol, and it don't look like a lot, but it really is, the acceptance of alcohol is down. And look at this, when you look at a DBA mouse, it's just like a DBA mouse. So we took an animal that genetically loves alcohol, right? And we converted it to an animal that hates alcohol by giving something called diphenylalanine, which in a sense raises the level of endorphins, which we studied. Now, what we have to realize is that dopamine is very important. And what happens is that if you have a low dopamine function, people will self-medicate to get dopamine release. Now, this is a demonstration of functional magnetic resonance imaging in the brain following food, music, and cocaine. And we're looking at different levels of how powerful these three agents are. So, for example, food gives you a 6% release of dopamine in the caudate region of the brain. Music gives you 9%, by the way. And cocaine is 22%, and sex is even higher, okay? I took out one of the slides because we only got 15 minutes, okay? I think you're not interested in sex. At my age, yeah, I don't know about it. Okay. The common neurochemical mechanisms between drugs and non-drug addictive behaviors are opiate antagonists like naloxone block alcohol effects. Lower brain endorphins result in higher alcohol drinking. Raising brain endorphins by preventing its breakdown reduces alcohol drinking. All addictive behavior, drug and non-drug, cause the acute release of brain dopamine, and that is very important. That is what the person, the patient, is seeking, especially if they're born, let's say genetically, with a dysfunction of the dopamine system in the brain, right? Okay, so now let's talk about, see where we are at this point. So what is genetic testing in the addiction field? All right? So we're talking about single nucleotide polymorphisms. So genes code for proteins that can be structured or metabolic, hundreds of thousands of molecules long. Okay, either adenine, guanine, tyrosine, cytosine molecules, and each site is called a nucleotide. Okay, a singular nucleotide polymorphism is a substitution, okay, of one site. For the most part, genetic testing identifies normal versus abnormal segments. It's really rather simple. Okay, so if you take a look at that, it's the normal sequence when you get a substitution, you know, you get an A to a T and it goes to a G to C, bingo. Now you got a molecular rearrangement and you no longer get the effect where the gene is doing its job. It's lost its activity, or it may have made some other activity, okay? So that's very, very important in understanding genetics. So pharmacogenetics predicts metabolism of prescribed drugs. Now you might have seen that there's companies out there now comparing SNPs on liver enzymes to metabolic rate for targeted drugs. Slow metabolizers need less drug, or if active metabolite, the drug won't work, and rapid metabolizers need more drug, okay? So there's certain genes that can be measured, and you use drugs, you can determine whether or not you need a higher or lower dose during treatment. Now that's just one aspect of genetic testing. What we're really going to focus on is something a little bit more. We're going to focus on something that has to do with genomic testing. And the basic idea is that you can genotype, you identify the allele, okay, using what we call the genetic addiction risk score. You can target the polymorphism, okay? And then you can actually develop even customized products that you target against the actual allele of risk, okay? And that affects, of course, you're looking for an effect to it, to bring about a balanced dopamine, okay? And you end up with potentially a nutrigenomic solution in terms of what you're looking at. Now that is the ideal. That's what we're trying to do. So what we have done is that we've looked at something called the genetic addiction risk score, which we developed. And what this does is examines the combined sums of many related polymorphisms to determine genetic risk. It is the sum of the number of risk alleles as measured by a panel of genes rather than any one gene alone. Any one gene alone does not have enough contribution to the variance. This genetic addiction risk score, GARS, is based on functional polymorphisms of reward genes. It's possible to find significant association with the degree of risk for all addictive behaviors, or RDS. So it turns out, way before that, in 1990, in my lab at the University of Texas worked with Ernie Noble at UCLA. And if you can remember, in 1990, we published in JAMA the very first psychiatric genetic study. And what we showed was that on chromosome 11, okay, in Q22, there was something called the dopamine D2 receptor gene. And what we were able to find was that if you're basically an alcoholic, okay, you have an extra band. It's called the A1 allele. And if you're non-alcoholic, you don't have that. And we're able to associate as high as almost 80%. And these are actually people that died from alcoholism. So we look at brain tissue compared to controls brain tissue. Now, it wasn't perfect, okay? But because we found out 20% of controls had it. But the point is that this was the first demonstration that you could use this technique, it's called RIFLIP, to actually study genes. Now, there are so many studies from that time on. The important thing was that once we found this genetic association, we were able to now look at the binding, okay, and carries these are all the brain tissue of humans, and we're able to show that if you carry the A1 form of this gene, you have 30 to 40% less dopamine D2 receptors compared to the A2. So you have really a reduced number of dopamine D2 receptors. And you need these receptors, of course, for dopamine to work. If you don't have these receptors, dopamine's not going to work, right? Makes sense. But now look at this. Guess what? 100 million people in the United States got this A1 form. Now, when we first did this, they thought we were crazy. Because how could you have a disease that is 100 million people in the United States? It's like, it's all of California, you know what I mean? Or more. So, you know, so they thought that, I mean, the controversy, I had to live through it, okay? I can't go on that, because that would take another 40 minutes to tell you how that went through, okay? But just take a look at it. It also has different associations with different ethnic groups. For example, the African American has it at 50%, one in every two. The Hispanics have it at 58%. The Asians at 72%. And you say, well, Asians, they don't drink alcohol. Well, they can, because they get flushed, but they have all the other addictions, even photographs, okay? Or gambling, you go to Las Vegas. And 85% of the American Indians. So that's like an aha moment. Ah, what? Okay, so that you can see that it's an amazing array of association just in one gene. Now, so we have developed in the last 25 years, from that time, something we now call the genetic addiction risk score. So what we now are talking about is a panel of genes, okay? And the panel is D1, D2, D3, D4, dopamine transporter, serotonin transporter, mu-opioid receptor, GABA B receptor, monoamine oxidase, catechol methyltransferase, and we hook in the cytochrome P450. And that is called the genetic addiction risk score. So what we're really saying is that when this hits the market, what we're going to be able to do is, at birth, see if a child has risk for addiction. Now, you can look at me and you can say, my God, you're labeling, and that's not good. However, the good news is that there may be something to help that individual, okay? So we have to take a look at that. So what we do to do this is that we do a DNA collection by saliva, okay? And you just saliva, and then we do genotyping, okay? And we measure, what we measure is this. We only measure the risk alleles, okay? And we count the number of alleles that you have. In this particular panel, we got 22 risk alleles, because you got 10 genes. You actually got, for male you got 22, for females you got 21, because the X and the Y. But what I want to show you, and this is only since 2014, it's even more now, but what this shows you is studies that have been done with these risk alleles. You know, that's up to 4,000 or more, okay? So you can see that you got thousands of studies to show the association of this risk allele for all kinds of RDS behaviors, okay? Now, so what we did, I'm sorry you can't see that slide well, it's supposed to be a different color. So what we did is we did an association study, and we compared the GARS against the Addiction Severity Index, and that's mandated in about 20 states. And we evaluated 393 people from 8 treatment centers across the United States. We did the genetic analysis, which was performed independently at the Institute of Behavioral Genetics in Boulder, Colorado. And then we took a subset of 277 of these people, and they took the ASI alcohol severity index and the drug severity index. And then we studied them, okay? Let's see what happens. So what we found is different percentages. So if you have 6 or less risk alleles, 28.7% had that, 7 or 8, 30.9 had that, 9 or higher, 40% had that. The interesting thing is, since these people were in treatment centers, every single one of them, 100%, carried at least one allele, okay? Now, here is the interesting thing, okay? So when you do chi-square analysis, mixed gender, with 7 risks, we can significantly reduce alcohol severity, okay? At the point of 0.04, okay? Age, when we control for age, we also get significance. What we're saying to you is this: if you do this test and you show that that individual has 7 risk alleles out of 22, you can predict alcohol severity in that individual. Now, if the illegal drugs is something else. I'm going to tell you that. I took this slide. In illegal drugs, if you have 4 alleles, you can predict drug severity. Because remember, illegal drugs are pretty more powerful, okay? So you don't need as many alleles to get the effect in bango. You know, that is the problem of the opiate epidemic. Because what we're seeing is this: people are coming into pain clinics and they're gonna get what? Oxycontin, bango. And 100 million people are walking around with the risk of just one gene. Now what do you got? Now you got a mess. Now you got people coming and getting oxycontin and say, "Oh, gee, I love this." Oh, of course you love it. You got 4 alleles, you should love it, you know? So that is your problem of the opiate epidemic. So the summary of this particular study shows that in 277 people, you got low risk, moderate risk, high risk, the alcohol severity, even age 0.012, the drug severity, we found that at 0.05. And I think the reason why we got less strength for the p-value in drugs is when they take this test, we think they're lying. People don't want to tell them what they're doing. And in fact, if you carry the A1 allele of the dopamine due to receptor gene, you have a higher chance of lying. So there you go. I rest my case on that one, okay? Now, can genes predict relapse in clinical outcomes? That's important, right? You want to know if they can predict it. Well, it turns out that they could. This is one example. So an example of the impact of genetics on recovery. Relapse. Carriers of the dopamine D2A1 allele are significantly at a higher risk for drug relapse than carriers of the D2A2 allele, which has the normal complement of the D2 receptors. But look at this. What are medical problems? So carriers of the D2A1 allele have a higher rate of both hospitalization and mortality. And it's a 10-year follow-up studies, okay? So that's not too good, all right? Now, what about gene therapy? So since we went through all this, can you go ahead and do gene therapy and do something that you can increase the D2 receptors in the brain? And this was done, Dennis and Noah Volkman, we did this with mice, we did it with animals drinking, and what you do is very interesting. You actually insert, okay, and that's in the image this was the rat brain, that show an increased expression of dopamine D2 receptors in the nucleus accumbens. So when you insert the cDNA of the DRD2, what happens is to start making more dopamine receptors. Now, the left side was injected with the D2 gene compared with the right side, which was not injected with the receptor. And I don't know if you can see it that well. But the point of the matter is that when you do this, guess what? The number of the dopamine receptors increase. Now, when you increase the number of the dopamine receptors by doing this gene therapy, which is something that we do directly into the brain, and we're not going to do that in humans, although I can tell you, in China, guess what they're doing? They're obliterating the nucleus accumbens. Well, we're not saying we're going to do this, but you know, it's better than obliterating the nucleus accumbens. And they've done that on 400 people, military people. But we're not doing that. This is just to show that Now what happens when you do this theoretically, the dopamine receptors go up, and that's this yellow, right? They go up, and the drinking goes down, and we did it in cocaine addicts, and cocaine, this animal self-administration, goes down as well. So raising dopamine D2 receptors can reduce alcohol and cocaine-seeking behavior, right? That's just theoretical, okay? So we have to worry about epigenetics. We have to worry about how the environment is affecting the chromatin structure on top of the DNA. The DNA is not affected at all, right? That's permanent. But epigenetics has an effect onto the histones sticking out above the DNA, okay? So when you look at this, if you get methylation, you block gene expression. When you're old, that might be great, but you need to have your genes working. Otherwise, in certain areas, especially in the reward center, if you deacetylate, it unfurls the gene and allows for the expression of that gene. It's as simple as that. So, one of the experiments was done at Mount Sinai, and it says, walking around, where's my dopamine? Okay, I don't have my dopamine, what's going on? And then you want your dopamine. So, what the lessons that you learn from this is that using a genetic technique called antisense, okay, you can knock out a specific gene. And one experiment, scientists knocked out the DRD2 gene in mice, and then they bred future generations. They found that compared to normal mice, the DRD2 negative mice showed two interesting results. A, when administered morphine chronically and then withdrawn from that substance, the resulting withdrawal symptoms were the same in both normal and knockout mice. However, when tested for reward, the fairly good response for morphine administered to the knockout DRD2 negative mice, they had no rewarding or euphoric effects from the morphine, while normal mice showed expected reward behavior. So there's no reward, there's no euphoria, you're not got dopamine, okay? But, look at this. I'll do anything to get heroin, okay? Now what does that mean? Well, parental mice were injected with marijuana, the active ingredient in cannabis, well, THC. After two generations, it was found that the DRD2 gene had a high rate of chromatin methylation. After 2 generations, it was found that the DRD2 gene, the messenger RNA expression, was significantly reduced, okay, compared to normal mice. After 2 generations, it was found that the F2 generation siblings had a high sensitivity to what? Heroin. So you give a marijuana to a parental animal, and then give it birth, and the next generation, and then the next generation, you check it again. Now all of a sudden, these guys love heroin. So that's called epigenetics. Now, the question is, can the recovery environment be used to repair genetic and epigenetic brain dysfunctions that predispose addictive obsessive compulsive behaviors, or what we call RDS? All right. The main thing is, you gotta love your pups. If pups are abused, or people are abused, guess what? After 2 generations, that abuse is causing methylation on very important genes. So that whole idea that you gotta love your children is now coming into the front. We're beginning to understand that, right? All right, so you love your pups. Now, are there ways that you could treat patients and boost up your dopamine? Well, there's things like yoga. There's dopamine boosting foods. There's a book that I just did with Malibu chefs, with Joan Boston, called Dopamine for Dinner. Meditation. You get a 65% increase of dopamine. Spiritual practices, trauma therapy, exercise, right away, 12 steps, and you'll see later, we're talking about neurofeedback, okay? Now, is there any genetically based treatment that can induce molecular epigenetic changes and improve dopamine homeostasis? My friend John Coward don't like the word dopamine homeostasis. I like it, I'm using it, okay? Now, the brain reward cascade. So we talked about that earlier. Gene targets in the brain reward cascade, and if you notice, you know, serotonin and caffeines, and we have the opiate receptors, and it's affecting GABA and the glutamate, and you're getting down to reward. And if you look at the other side, you got a whole bunch of genes that are actually affecting this whole system, okay? And if the genes have polymorphisms, then you're changing that system. So when I say that dopamine is important, well, so is serotonin. So are the endorphins, right? So is glutamine. So is GABA, okay? So it's all interactive, it's a cascade. Now, so over the last 40 years, we have developed something called the new adaptogen amino acid therapy. It's a grass, and it contains D-phenylalanine, L-phenylalanine, L-tryptophan, L-tyrosine, L-glutamine, chromium, rhodiola, and pyridoxine. Now we have a new variant. We're putting N-acetylcysteine, NADHD is involved, and Q10. But notice that what we're really doing is affecting the opioid system, the dopamine system due to synthesis, the serotonin system, again the dopamine, the GABA system, chromium affects serotonin. Rhodiola not only does it block catechol-methyltransferase in the synapse, it blocks monoamine oxidase when the dopamine comes back through the transmitter into the pre-neuron, right? It goes into the mitochondria. Monoamine oxidase eats it up. We block that. So we're pushing the dopamine system. Now you can argue, are we getting too much dopamine? But we'll talk about that a little bit. Now, this is showing some of the effects. This is healthy volunteers, and increases focus, okay? This is after 30 days of treatment. They look like this. Well, this thing doesn't. This, and then you see that, look at that. Increases focus. What else does it do? Well, there's some other thing. Certainly can reduce craving. This shows stress is measured by skin conduction, placebo versus KB22, and it reduces stress, okay? So it reduces stress. Okay, now here's very interesting. PTSD. People with PTSD have terrible, some of them, nightmares, okay? We took 13 people, we put them on this, and guess what happened? We knocked out nightmares, okay? And the interesting thing is that we have another paper that we showed 4 of these people, their nightmares are gone even after 12 months without using it. So there's some kind of neuroplasticity going on, okay? Now we gotta come to the functional magnetic resonance imaging. So we wanted to see if this compound, KB220Z, can activate brain sites, you know, by using functional magnetic resonance imaging. So we looked at rats. This is done at the University of Florida, and you can see the rat going into a magnetic. It's a couple $1 million magnetic field that does functional magnetic resonance imaging. We looked at over 65 brain regions. That's a heat map, and I don't, you know, you could ask me questions, but that's a heat map, and you can actually see what's being lit up and not, but not everything's being lit up. Now this is just one example. You see the region, you see the nucleus accumbens, and bango, what happens is you can see that placebo and the nucleus accumbens are doing not very much, but look at that. That's 5 to 15 minutes after you orally given the KB220Z, and you can see that that's dopamine. All that orange is bold, that's dopamine. It's been proven, okay? So dopamine is activated. This shows when you see one part of the brain, you get significant differences in the other part of the brain. So it just shows that it's selective for certain areas. Remember, 65 areas, and these are the only ones that show significance. So it's selective to the reward system in other parts of the brain, like cognition. Now this is interesting. This slide shows that we look at something called connectivity volume. What connectivity volume means is that we're measuring neuronal recruitment. So for some reason, this substance, KB220Z, is recruiting extra dopamine neurons in areas, in the very important areas in the brain, so that we actually increase the neurons of dopamine firing in certain parts of the reward center of the brain. And that's what this slide shows. Now, then we did it in humans. In humans, we did this in abstinent heroin addicts. In abstinent heroin addicts, what you can see over here is this is placebo, that's the caudate nucleus accumbens. Dopamine is not even there, okay? But when you look over here, you can't see it that good, but dopamine, after 1 hour of KB220Z crossover experiment, you see that it's coming out. Sorry that you can't see it that well. But look at this, the retainment, where there's some emotional area, learning area, some motor area is reduced. We call that homeostasis. We're doing something to balance the brain. Now, in abstinent heroin addicts, and Joe Lubar was involved with the study, what we did is we looked at a QEG, and what do we see? We had abstinent heroin addicts, and when we gave the KB220Z and we looked at the singular gyrus, okay, we found that you have increased alpha and we also had increased low beta. And if you remember, Joe, you said that you get that like in 10 to 20 trials, and we got it about in an hour. So think about the combination with neurofeedback. It might be very interesting. So craving is one side and relapse is the other side, because we're blocked. The singular gyrus is the site for primary relapse, okay? All right, we gotta move on. So the summary is, naive rats, significant increase in resting state, function connectivity, very important. Naive rats, significant increase in connectivity volume, very important. Human abstinent, psychostimulant addicts, QEG, we block widespread theta. In the singular gyrus, increase alpha, increase low beta. Human abstinent heroin addicts, increase resting state function activation. At the nucleus accumbens, enhanced connectivity volume. In rats and in humans, okay? Now, then we did some Loretta work with ADHD with Bruce Steinberg. And this is a scan of an ADHD guy, and you can take a look at the scan, of course, theta, alpha, beta, and high beta. Now, I don't know how good your eyes are, but the theta actually went up, and we show that working memory increases. And the alpha went up, and the low beta went up, and the high beta went down. And you can see that, okay? And Joel is involved with that paper as well, right, Joel? Okay, now by the way, we're doing IRB, we're getting 10 ADHD guys to compete. Okay. Now, I got a couple of minutes, and I know you're standing there. So we are proposing the reward deficiency solution system. So it consists of the genetic addiction risk score, a comprehensive analysis of reported drugs, which I didn't talk about. That's drug testing. Dopamine agonist therapy, KB220Z, future pre and post RNA analysis of genes during treatment, many holistic psychological things that you can put in to boost dopamine. We'll talk also about Eureka as one example, neurofeedback to enhance happiness and cognition via dopamine regulation. And we're trying to induce what we call dopamine homeostasis. Now, basically, we're talking about, say, neurofeedback, say Eureka. We know that from FOI studies that pleasurable experience excite the medial frontal cortex. They include winning money, other pleasures. This is prefrontal end of the dopamine system. It's the highest concentration of dopamine neurons in the cortex. The pig brain happiness trainer, or Eureka, activates sites near the FPZ is over the prefrontal terminus of the dopamine reward system. Now, so Eureka is a way of processing EEG that can be used to measure and train the brain system for processing new learning and discoveries. Eureka is a short for neural Eureka. New discoveries make you feel more aware, happy, satisfied, where they enhance your learning and memory. Dopamine, of course, enhances memory consolidation. It is a clarification of 40 hertz gamma EEG rhythm involved in memory and learning. The brain rhythm from the central thalamus scans the cortex and sends info back there to be understood. It stimulates the prefrontal pleasure center to reward and so forth. If you're doing that, you may get dopamine release. We don't know that yet, but what you have to start thinking about is applying functional magnetic resonance imaging when you're doing neurofeedback to really see what's going on and see if you're activating dopaminergic sites. And finally, happiness. In other words, if you're taking this Israeli group of I don't know how many thousands of people, that's the basic happiness. When you use the Eureka, you see that the happiness goes up, and 4 months later, it's still up. So there may be something going on for neuroplasticity that you're getting, say, even with neurofeedback. So basically, we're saying this, that happiness equals dopamine. And as you can see, people are tattooing happinesses all over themselves. And I think, instead of saying, how you doing, I think you say, how you dopamine. All right? So, and that's all my people. Now, if you just put this one thing on, I know we gotta get out, and could you put that song on? Could you hurry up and then we'll be finished, okay? I wrote a song in Austin with some heavy cats on dopamine. You gotta hear it, and I hope that speaker don't shoot. Please put that on, okay? And thank you. I hope you like it. Don't leave, you don't leave. You gotta hit the dopamine, so get back. Shut the doors. Okay. He said he could do it, so hope he can do it. I wrote the song here. I wrote the words, the lyrics. If he could ever get it on, I hope not, and I'll sit back here so you can see it. I hope you get it on. Come on, Zach. All right, make it loud.